Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. Today we will discuss about the glycogenolysis. The meaning of glycogenolysis, the breakdown of glycogen into the glucose. So the glucose formation from glycogen. So here is the human which contain a digestive system. So first of all, we should understand about the digestive system is here. So here's the liver, which that is the important for the glycogenolysis from the glycogen, or on the other hand the muscle. So here is the stomach, which that is the digestive part of the human body, and here is the digestive system contain. Stomach contain pancreas, which that is important for understanding the glycogenolysis. So here is the pancreas, which contain a different type of cells is known as pancreatic acini. So these pancreatic acini are island of Langerhans. It's also known as produce endocrine hormone glucagon, insulin, and somatostatin. On the other hand, the exocrine also activity in the pancreas to produce amylase, peptidase, and basically amylase, pancreatic amylase, and pancreatic lipase. Trypsin and chymotrypsin is also produced. So here is the pancreas, which that is contain island of Langerhans, which contain a beta cells. So beta cell produce insulin, and alpha cell produce glucagon. And here is the green color shows that the beta cell, and alpha cell is produce glucagon is showing in the white color. On the other hand, the delta cell will produce somatostatin, which that is used for the growth. And so that is why it is very important gland for the anabolic and catabolic activity. So here is I am saying that here is the glucagon release from the pancreas of the alpha cell to produce to release the glucose from the glycogen. How? So we will discuss about in this video. So here is the exercise, fasting, active working, a fear, rage, angry. These all activities increase the metabolism and increase the glucagon to increase the glucose level into the blood. On the other hand, adrenaline is also important for the releasing of glucose for the more active, alert, and consciousness. So the important thing is that here is all thing is related to the production of glucose from the glycogen, that is known as the catabolism. If we will talk about the anabolism, so the anabolism mean the glucose will form into the glycogen, so that is known as anabolism. So this catabolism and anabolism is collectively called is the metabolism. So here we will discuss about the catabolism. So the catabolism is the breakdown of glycogen into the glucose. So here I am drawing that exercise and running is also impact to increase the glucose from glycogen, is known as glycogenolysis. And the next video we will discuss about the glyconeogenesis. So here first of all, here is the gallbladder, which contain liver, and that is used for the making a bile acid, but here is the stomach and that's the liver. So this is the digestive system, and digestive system contain liver. So here we will discuss about just liver, and the liver is the largest part of the glandular organ in the human body, which contain two lobes, left and right. One is right and one is left. Right is smaller and left is larger. So here I am drawing the blood vessel which contain a blood. So it means it's also contain a hormone. For example, the adrenal gland will also produce the adrenaline, cortisol, epinephrine, norepinephrine. So here we will discuss about just adrenaline. So the adrenaline and glucagon will release into the blood, and that stimulate the liver cell, muscle cell, and liver cell and muscle cell, which that is for using to break down the glycogen into the glucose. So the glycogen into the glucose is the formation. So here we will discuss with the detail. So here is the muscle and liver. Liver contain a bilobed organ, is very important metabolic central part of the human body, which is the breakdown and making something new, which that's anabolic and catabolic activity into the liver. So here I am drawing that is a glucagon receptor or adrenaline receptor. So you should suppose that is the glucagon receptor, which contain three subunit of protein, which making alpha, beta, and delta. So that's used it is present the GDP glycerate diphosphate, no sorry guanosine diphosphate, which that's activate by the GTP to GDP conversion, and this conversion to make a GTP, and this GTP is separate from these two subunits. So here is I am drawing again glucagon receptor that's go far basically here, and that is the GTP again will be produced by the ATP to convert again GDP. This also process is can be acceptable. So here is the alpha GTP will be separate containing protein, and these adenylate cyclase enzyme pass to ATP to convert cyclic AMP. So that is the GTP. Alpha GTP is the trigger to release. That is the primary stimulator, GTP, alpha GTP, which that stimulate to produce ATP to cyclic AMP. So this cyclic AMP will go with the activate to protein kinase enzyme. So this protein kinase enzyme will be activate and phosphorylate protein kinase form, and this formation will be stimulate the PPK. It means the phosphorylated phosphorylase kinase, and this phosphorylated phosphorylase kinase will be activated by the ATP using to produce ADP to convert phosphorylated phosphorylase kinase phosphate. So this kinase phosphate is attached the phosphate group, and this will be stimulated by the glycogen phosphorylase. So this glycogen phosphorylase enzyme PYGB is stimulated by ATP using to produce PYGA, that is known as glycogen phosphorylase A, and that is the phosphorylated. So here I am drawing the glycogen. The glycogen is the branched chain of polypeptide of polysaccharides, that is known as a polysaccharide, and that's contain alpha 1 to 4 glycosidic bond as well as alpha 1 to basically alpha 1 to 4 and alpha 1 to 6 glycosidic bond. So here is the glucose 1 phosphate will be formed from the glycogen by the separation. So the glycogen phosphorylase A will be separate glucose 1 phosphate. So here is the important thing is UTP. That's a UTP mean uridine triphosphate. So the triphosphate three molecular phosphate alpha, beta, and delta will be bind with the glucose, and here is the structure you can see, and that's convert by the using of glucose to glucose, that is using to glucose to produce UDP glucose. So the two pyrophosphate, it means pyrophosphate will be released and form a UDP G, basically uridine diphosphate glucose. So this uridine diphosphate glucose will be further processed, but first of all we will discuss is the backward reaction during the fat state. So these all thing is during the fasting state are more active during the sympathetic nervous system. So that is important thing is here is the GYS, basically GYS A is used by the using of ATP to convert GYSP phosphate. So the phosphate group will be attached. So these attach to activate the glycogen formation. So this glycogen formation will be occur. So again, it is reversible reaction. It is not irreversible reaction. But here we will discuss just the glucose formation. So that this glucose is contain phosphate group. So we should separate the phosphate group from the glucose to release into the blood for increasing the glucose level, for using further into the cell, or remain into the blood for increasing the glucose. So here is the phosphate group attached, is known as the glucose 6 phosphate, which that is using the enzyme glucose 6 phosphate by releasing the phosphate group to make a whole complete original glucose molecule. So this original glucose molecule is transported by the glucose transporter 2. This glucose transporter is used by the active transport by the two sodium ion move outside and one potassium ion move inside to release it into the blood. After the releasing of blood, that means glucose level will be high, and that's why glucose level will be high, that on the basis of requirement. So first of all, we will discuss about these glucose molecules are present into the hepatocyte of the liver cell. So that can be enter into the glycolysis. Glyco mean sugar, and lysis mean breakdown, to produce 3 molecule of pyruvate of 2, and these will be converted into the acetyl-CoA into the mitochondria, and that acetyl-CoA will be released into the cytoplasm of the hepatocyte muscle cell, and these will be again ATP, and these ATP used to produce more glycogenolysis, the breakdown of glucose more. So this ATP can be used from here, or go the complete sugar molecule into the blood. So what happens when glucose will be go into the blood? So sodium ion will be moved into the blood is also because it's here. So that is glucose enter into the bloodstream. So here is the island of Langerhans produce the glucagon from the green color, shows that the beta cells of the pancreas. So that insulin will be produced from the pancreas to it means the increase the on the signal of the increasing of the glucose to create more sugar. That's more sugar will be created, and that's created sugar will be go into the muscle tissue or something where the need. So the on the basis of need that is required, so the glucose transporter which will be allowed by the binding of insulin from the insulin receptor into the muscle cell. So these insulin receptor will be facilitate to enter the glucose into the cell of tissue, for example muscle cell or myocytes, for create energy. So the muscle contraction will be occur, or something which that is metabolic activity will be occur. So that's all thing is about the glycogenolysis. So thanks for watching. Please subscribe the channel.